Epigenetics is a study of chromatin modifications that affect gene expression without altering DNA nucleotide sequences. Epigenetic modifications, including histone modification and DNA methylation, are established during early development and are maintained during cell division. In cancer, in addition to genetic changes, epigenetic patterns are also altered and disrupted. For example, there is simultaneous genome-wide hypomethylation and hypermethylation of promoter regions that lead to alterations in gene expression. DNA hypermethylation of promoter regions is associated with silenced genes, including silencing of tumor suppressor genes. And gene promoter hypomethylation is associated with activation of genes, including activation of oncogenes. Altered gene expression of tumor suppressor genes and oncogenes is present in all tumors, allowing the emergence of different abilities and characteristics of cancer cells, such as increased cell proliferation, matrix degradation, cell motility, immune system evasion, and a capability to metastasize. In cancer, epigenetic silencing of tumor suppressor genes is well established. Let us look at colon cancer as an example. Here we have colon cancer cells. In colon cancer, hypermethylation of the promoter region of the MLH1 gene, which is a mismatch repair gene, is common. Methylation of the MLH1 gene promoter is associated with silencing of the MLH1 gene. Because MLH1 is not expressed, MLH1 is unable to repair mutations in colon cancer cells, resulting in increased cancer genetic lesions. Scientists can now determine methylation alterations or signatures in an individual's genome. Determining the DNA methylation pattern in the cancer provides a new biomarker for early detection and for identifying the cancer type, pathogenesis, and progression. Scientists can now use the unique DNA methylation patterns as a source of biomarkers in individual cancer patients, and this can allow for early detection and diagnosis of cancer type, monitoring the tumor progression, and predicting treatment response and outcome. Let us first see how biomarkers can be used for early detection and diagnosis of cancer. Epigenetic changes, including hypermethylation of tumor suppressor gene promoters, occur early in tumor growth. In addition to analysis of tumor tissue DNA, many tumors shed cancer cells and cancer DNA into the blood and other bodily fluids, including stool, urine, and saliva. Body fluids therefore provides a non-invasive source of cancer DNA that could be potentially used for early detection of cancer by the sensitive detection of aberrant DNA methylation biomarkers. For example, stool DNA with methylation of the MLH1 gene can be a potential epigenetic biomarker for colon cancer. And in sputum, promoter methylation of gene P16, a tumor suppressor gene, is a potential epigenetic biomarker for detection of lung cancer. In urine, hypermethylation of the glutathione S-transferase gene is one of the best epigenetic biomarkers for prostate cancer detection. Biomarkers can also be used for monitoring tumor progression. Each cancer is unique, and the epigenetic patterns continue to change as the tumor progresses. For example, here is a DNA of a cancer cell. As a cancer cell progresses, so does the alterations in the DNA methylation pattern. Monitoring the methylation pattern of critical genes promises to provide accurate determinations of the stage the cancer is in. As well, the methylation pattern could also provide predictive information of how the cancer responds to treatment. For example, in breast cancer, women with estrogen receptor positive disease are given endocrine therapy. But many cancers develop resistance, and so the methylation patterns stay the same. The methylation of cancer DNA can therefore be used as a potential biomarker to determine if women should be placed on different therapy regimes.